So landmark study, but there's a lot of background that you've got to have to really understand this study and really get the most out of it. So let's get into it and take a journey into the oral microbiome, this incredibly complicated ecosystem uh, within the mouth, this vibrant community of organisms that play a role not just in oral health, but we're beginning to see in systemic health and seeing concrete ways that they affect systemic health. So first of all, you got to understand it really is an ecosystem. There's over 700 species of bacteria. There's fungi, which can actually communicate with the bacteria in certain instances. Viruses, protozoa, which are like large bacteria cells that are more developed and even more pathologic in many cases. And there really are billions of them, and they're highly structured, so it's incredibly complicated. And then each one of those may have many types of subspecies that may have wildly different qualities, abilities, and they can swap different information between the two. So it's an astronomically complicated thing. Now, the mouth really has perfect conditions. It's the right temperature, it's moist, it has a ready food supply. So it really takes off and really only second to the gut in terms of bacteria within the body. Again, it's incredibly high. So you got to think of it. It's not a solitary entity. It really is a planet, an ecosystem compared to any sort of forest you would see outside. So now you got it because it's so complicated. You have to try to abstract this complexity and try to explain it. And one way they try to organize types of bacteria, life in the mouth, is based on if it's symbiotic, meaning it helps us. And there's some evidence that various bacteria in the mouth actually helps break down nitrogen. There's some bacteria species in the mouth that can actually interrupt corium sensing, make other types of bacteria less structured and can compete with sort of fast growing bacteria. So those might be symbiotic, and certainly the gut has many symbiotic ones. And many types of bacterial organisms, microorganisms, aren't really good or bad, they just live. And those are commensal. And then there's pathogenic ones that can interfere with our immune cells, cause tissue damage, cause inflammation, and certainly this type of bacteria we're going to be focusing on soon is in that category. But there's many others. Another way is to think about it as a core microbiome, where the average person has a large amount of bacteria. And certain things that person may do may predispose them to other types of variable microbiome. That's based if they smoke, if they have low saliva due to medications, if they have too much sugar, too much acid, vaping. All these things can change your microbiome one way or the other. In some cases, can actually make it less diverse and more pathologic. Now, the idea, and this is a very broad generalization, but it is an attempt to try to understand the complexity, is you may have a symbiotic sense of organisms in your mouth that don't really cause cavities, don't cause periodontal disease, don't seem to cause any problems, but are still there. And then there's some that are dysbiotic that cause disease. They start to grow uncontrollably. And this is what I see as a dentist. This is what I see very often. So you got to understand there is a landscape where these things are incredibly highly evolved. You know, the dinosaurs had periodontal disease. So ever since we've been eating food and there's been stuff in our mouths the bacteria can feed on, there's been issues. So they've been evolving much longer than we have. And they're very adapted to staying on hard surfaces. And as our diet changes, the types of bacteria they eat that survive change as well. So many bacteria are highly adapted to live on teeth. And we're talking about calculus is very sticky, and calculus can actually be made by bacteria as well, or contribute to plaque formation, which is then becomes calcified. Strep mutans drives caries. So it's the same idea. You can still get a home for bacteria that actually drives into the tooth, but it's the same concept where they're adapted to live in the tooth, or they could be adapted to live in the soft tissue around the tooth, the gums, the cheek. Even they can live on the tongue. They can live within the deep crypts around the tooth in periodontal disease, and that rapidly changes your profile. And what guides the two are many factors: the acidity of your saliva, your diet, if you smoke, all kinds of different pathologic factors. Now, there's a lot of specialization, there's a lot of communication, and different types of bacteria and teams of different bacteria are adapted for different types of surfaces. Now, it is a biofilm. You can think of it as a city where it is highly structured, and the longer it sits on the teeth, the more structured it gets. And it's not just food, it really is a living structure. And these bacteria can communicate with each other and achieve, again, more degrees of complexity. And it's not just periodontal disease that gets that, grow these sort of broccoli-like structures. You can actually look at fractal index of cavities as well, how highly ordered they are. And this multi-species community starts to emerge, and certain types of bacteria are start to be seen in more advanced disease very predictably. So there is a foundation. This is something a lot of dentists learn in school, and this is just the beginning of the story. Now, there's an acquired pellicle. After you brush your teeth, you have a smooth tooth surface, and various proteins, glycoproteins, are going to immediately stick to it. This is called the acquired pellicle. And this stickiness, especially if it's from the saliva, may have some protective qualities in some instances, but what's called pioneer species, Streptococcus is most widely known, will begin to stick to that. Now, immediately, there is, or very soon, there are secondary colonizers, which will stick to those bacteria, and that is how plaque begins to grow. And they start to form a matrix, this extracellular matrix from polysaccharides. And polysaccharides are derived from food in our diets, sugars and stuff like that. And they form this glue-like substance that also serves as a food source later on. So they can eat it, and it also forms the plaque. So the others is once microbes receive a specific threshold, once they get to a certain threshold and there's enough of them, they begin to undergo corium sensing. And certain types of bacteria, like Poriformis gingivalis, are the generals. They run how different types of bacteria communicate, and they send certain pieces of information like their concentration gradients, so they can determine where they are, how they grow. They can react to antibiotics. They can do all kinds of stuff, and their structure begins to really take off. And if you inhibit corium sensing, it does just the opposite. So this is really key to how these organisms start to begin to see it as almost a single organism when it gets to a large enough size. And various things start to happen. As more food gets trapped, more bacteria starts to die. There's later forms of bacteria. There's more cleanup type bacteria, especially in periodontal disease, will start to feed off amino acids and stuff like that. And oxygen levels begin to drop. You have more anaerobic bacteria and stuff like that. So you can think of it as a fortress where bacteria are walling themselves off. They can even undergo programmed cell death to help build this structure even larger. And they become fundamentally different than their free-flowing counterparts. And they can shield themselves. So they're very much more resistant to antibiotics. I see people that do mouthwash all the time with severe periodontal disease, and it just often doesn't quite help. It's not enough to penetrate, and even if it does kill a few cells, now you just have more dead cells to deal with. And that is how periodontal disease and other types of infections in the mouth begin to propagate. And they just gradually grow and grow over time, and they don't really stop. So the notable ones we all talk about, strep mutans is most widely known as driving cavities. Poriformis gingivalis drives gum disease, and there's many, many others. And what the key difference between the two is based on your pH levels, your salivary levels, your diet. If you smoke, all these different things can dictate which one will do better. Now, there's been a lot of talk about the oral systemic connection. There's a lot of sort of snake oil about this. But again, there's more and more concrete evidence of how exactly these interactions begin to happen and how issues in the mouth don't necessarily stay in the mouth. And the difficulty is with this type of technology or these types of problems is the effect on the body of not brushing your teeth, not taking care, and not having a good diet um, will are probably stronger. Things that negatively affect the mouth negatively affect the body. So that connection is incredibly strong. So it's very hard to tease out what problems are being caused by the mouth and what problems are shared by the mouth and the body, if that makes sense. So, but there are some where we're beginning to see that there are problems that begin in the mouth and spread systemically. And a lot of them begin with the amount of bacteria that are in the mouth. These highly developed, highly structured bacteria that are incredibly evolved. And what happens is the mouth in many instances acts as a gateway. It is the gateway to the body. And certain pathogens can begin to exist there in huge, huge numbers, and they readily travel into the body, not just through the gum tissue. But this presentation is going to talk about a novel way that they can enter the body that isn't often discussed. Now, they can directly enter bacteremia. They can go directly through the gum tissue. In certain types of necrotizing periodontal disease, they can literally live in the gum tissue, and in periodontal disease, they can readily travel. You can see bacteremia in many, many different cases, even in healthy patients. After you brush your teeth, it can actually grow up. So it doesn't take much. Your gums are fairly thin. And there's been many, many connections where they can travel to distant sites, heart valves, infective carditis. I've seen even on healthy patients, and it's been traced back to oral bacteria. So this is a very real thing that can happen. There's a connection between rheumatoid arthritis, the amount of inflammation. There's a lot of talk about Alzheimer's disease and gingipans that are found both in gum tissue and there. And it's important to begin to understand these. Now, one way we always see, well, there's more inflammation in the mouth, inflammation must be bad. And that plays a role too, but not only are they causing inflammation within the mouth, but as they spread outside of the mouth, they cause inflammation wherever they go, and they cause damage wherever they go. Now, they have a far-reaching impact. And once again, associations are very difficult because anything that causes periodontal disease, smoking, obesity, everything bad for your health is probably bad for your gums. Every dietary issue, if you're having too much sugar, too much fat, whatever, it's bad for your mouth. So you're going to see a very strong association between the two. But to go further, you're going to have to do what they did in this 2024 Nature study where they actually show the connection. But we all know there's a connection. People that have cardiovascular disease often have periodontitis, stroke. It increases your risk. Your oral hygiene increases your overall survival odds ratio. And it's a two-way street, certainly, but that doesn't mean there aren't oral effects down the road. So it's, again, all-cause mortality. Problems in the mouth don't really stay in the mouth, and vice versa. So it really does raise the question, well, if they can interact with the heart, if they found some of these bacteria in tartar of the heart, tartar in the mouth, can they play a role in development of cancer? And this study certainly says yes, they can. And let's jump into part two. And this is a Fusobacterium nucleatum, which is sort of a long, thin bacteria that is incredibly sticky. It involves, and its primary use in the mouth is it allows late bacteria to stick to early bacteria. It just sticks to everything. And it is, very unique. It's not considered incredibly bad. It's an orange complex in the mouth. It's very much heightened in people with periodontal disease, but it's found in most everyone's mouths, and there's many different species of it. Again, it's the connector between the two. So it's, again, really a key bacteria. It's got various copies of genes that allow it to stick to other types of cells in the mouth. And this makes it incredibly useful. And when you look at it, it's a gram-negative bacterium, and these are just other ways to try to understand, what's going on in the mouth. It is anaerobic, so it doesn't survive in oxygen, so it does very well in the gut. It does well in periodontal disease in deep sort of pockets. It's long and thin, it's very pointy, and it's very different in shape. It has a very fundamental type shape. And it is that bridging organism. You can go from one to another. It can stick to almost everything. And it has many different varieties. And different varieties stick more, stick less, and have other types of qualities. It can actually pick up different types of genes with it. It doesn't have a set nucleus, a distinct nucleus. Its DNA sort of floats within, so it can pick up different abilities from other types of cells, such as those in the gut. And it provides structural stability for the biofilm. It's very important. And again, genetic material can be exchanged within it. And it really does allow for destructive pathogens to live in the mouth. And it continues to live in the mouth. So again, it is the city. They say it's the mortar that binds the city. And that's essentially what it is. And it's long been suspected that this has been found throughout the mouth. They find it within tumors. And the first one they found is colorectal cancer. It keeps popping up, and it's very common in the oral cavity. Most people have it to a degree, but it's incredibly rare in the gut. And if you sample anyone's mouth, you'll find various amounts of it within the mouth. It's very common, but again, incredibly uncommon within the colon. And you can find it in stools. They do stool testing, and they use the mouth as a control. But it's very much enriched in humans with colorectal tumors. And it's even found within the tumors themselves. And interestingly, the association isn't linear. It will occasionally spike up where you won't have any of it in your stool, and then occasionally, if you get sick or whatnot, it can begin, for whatever reason, it can begin to overgrow. And it will increase for a short time and then drop right off. That's something we see very, very commonly. But in some instances, it doesn't drop off. It's able to establish a foothold, and very often those patients are the ones with colorectal cancer. So the significance of it is F. nucleatum they're linked to more aggressive types of colorectal cancer as well. It increases the recurrence rate. It's very bad if you find it. It increases the rate of metastasis, and it decreases your overall survival rate. So it's not just a person of interest. It maybe is a causative factor. And when you look at how virulent these organisms in the mouth, how highly developed they are, it seems like it's able to live in the gut. It's not just there. It's not just a passenger. It's actually surviving there, and it's causing a severe immune response. Now, the real question is this a correlation of causation? And as we dive deeper into this paper, you're going to see that they really laid that to rest. There's really no chance. Is it a passenger? Is it opportunistic? Is it just finding it? And that may be part of it. Is it really initiating it itself? And they've leaded inconsistent results. Nobody's really been able to show if this is. But certainly there's a high degree of suspicion that Fusobacterium nucleatum, these bacteria in your mouth, are there to play. So really what they wanted to do is dive a little bit deeper. And that's exactly what this study did. So the true culprit, and let's dive into, there's a study, this 2024 study in Nature by Zepeda Rivera and colleagues looked at the genome of Fusobacterium and really told an entire story, and they also did some incredible animal testing, human testing, and were able to figure out and tell an incredible story about this. And one of the things they looked into is that, as there really was not a single entity. There's roughly 70 different species of Fusobacterium, and of particular interest, there are two clades, C1 and C2, and only one of them was found to be extremely strongly associated with cancer. There's a couple other subspecies that did pop up, and then they dive deeper, and by able to actually pinpoint the subspecies, they're able to make statistics a lot stronger, because not every type of Fusobacterium is always associated. So let's go to the next thing. So what they did, 55 tumor strains, and they isolated tumors. And, they found 55 tumor strains of this Fusobacterium, 80, oral strains of these from healthy individuals, and they sequenced them and determined exactly what was going on. And what they discovered is a Fusobacterium, or FNA, that by looking at the entire gene repertoire, there was, again, some bifurcation. They're able to tease out exactly what was going on here and dive into the genetic divergence of this. So even though they were mostly divergent, there was, certainly additional bacteria or different types of capability and duplicates of key types of genomes that allowed them to do different things. And again, by sequencing, they were able to really look which was more commonly found. And both of them were found in the oral cavity. Really was indiscriminate what type of bacteria, Fusobacteria, they're found. Now, FNAC2 topped out of all the ones. Every type of Fusobacterium is in the mouth, but only C2 is found in tumors. And again, there's a couple other kinds. And this is incredibly strong. The other ones don't. And why? Why was this sibling? Why was sometimes do, sometimes don't? What is the difference? And does it contribute? And what is so special about it? Why does it? What makes it live in the colon and the other ones not? And that really is the next part of this study. And when they looked at the genetic toolkit, they're able to find what tools it has. How can it survive? And it has this adaptive toolkit, a suite of genes that specifically allow them to survive the journey from the mouth to the gut. And not many genes can do this. And in some instances they can. You do occasionally see mouth Pulformus gingivalis, stuff like that, can survive in the gut and wreak havoc, but it's uncommon. The stomach does a very good job. So it has more genes. It has a larger accessory genome. Again, there's no set nucleus, so it's able to acquire additional pathologic qualities that likely came from the gut and fellow gut bacteria. So the key thing is it has a special system, GDAR, GDAR, that allows it to survive the acidity of the stomach, 1.5, which kills practically all bacteria. It will kill C1. So C2 has an advantage, and they're able to show different copies of it. Now, it's absent on C1, but they went more. They did laboratory studies, and they tested C1 versus C2. C1, of course, died very quickly when exposed to simulated stomach acid. C2 did not. And this really helps tell the story of, it lives in the mouth and it's continually sent down into the gut. And then, that begs the question, if it lives in the gut, it's spreading through a fecal route. Or is it spreading through a fecal route? Well, that's very possible. So, and they also need to eat. There's certain types of foods or certain types of foods that are found in the gut, not really found anywhere else. And surprise, surprise, C2 is able to eat them. It has them. And likely they picked it up from gut bacteria at some point. So the virulence factors, so it has an adhesion or multiple copies of it, so it's incredibly sticky. And it facilitates attachment to other types of microbes within it. And most interestingly, it has adopted um, various types of immune responses that are able to degrade host tissue. It's able to fight immune molecules. So it's able to basically pick up a weapon and start to not only stick to things in your gut, but start to fight your own immune system. And this is obviously incredibly bad. And of course, because this is a nature paper, they didn't stop there. They kept looking into its morphological differences, what it can do. Can it move? Can it colonize? Can it fight immune cells? And they found this was all a factor. So they kept digging. So it wasn't just, but they even looked at cohorts of patients and they wanted to see if C2 was found in real world patients, and sure enough, they analyzed tumors and they found that C2 was much more abundant than C1 within the tumors themselves, as if we didn't already know or suspect that. And they even paired samples, and they did find that total F-nucleotidim was higher in the tumors, but it was exclusively FNC2. No surprise, it's the one that specifically evolved to live in that situation. C1 is found. And interestingly, C2 is found in the mouth of these patients continually, but it's not extremely overgrown. It can live in the mouth, it's a reservoir in the mouth, but it's not explicitly evolved to live in the mouth. So it doesn't take off like some other types of bacteria. It's very specialized. They looked at it in stools, it was through the roof. No surprise in people with colorectal cancers. And again, the study didn't explicitly say this, but the question really is, well, hey, this is a bacteria that seems to do very well in the stool, in the gut. Is it transmissible within stool? If you're constantly pooping it out, does that mean you can then transmit it to somebody else through the oral fecal route? Is this an evolutionary adaptation where it can then continue to propagate to somebody else's gut? And certainly this is something many other types of bacteria do. Is this one doing it? I don't think so, but it makes sense that it might try to do that. So when you compare it, just to stress and F1 and FNA, Clade1, and again, there's many others. We're just focusing on Clade1. There's many others that do similar thing. Many are predominantly in the oral cavity, but FNA-C2 really is the clade that is found in tumors. And there are a couple of other subspecies that are suspect as well, but this was the one that was really jumped out. Now, of course, it's a nature paper. They kept going. So they built, incredible. You have all the circumstantial evidence, but you really need to see if it causes it. So what they did is animal studies. So they created a in vitro, and of course, they looked at it, and they showed in vitro tests as well, pro-tumor, and and it is literally can engage with some of the host cells within the tumor. And anytime you have something, an immune cell that, or at least a bacterial cell that grows very fast, grows any kind of structure like the ones you see in the mouth where it adopts some sort of fractal structure, you may expect a response from the immune system. And that immune system is going to have an effect on it in some way. And as demonstrated here, that is a negative effect. So the definitive experiment involved a mouse model where it was genetically predisposed to develop intestinal polyps. They had a control group, a placebo. They gave one group C1 orally, and they also were that orally gave the mouse C2. And it was a head-to-head study, so bacteria, and it was also a fusobacterium. So he was able to really eke out a lot of understanding from this. So the smoking gun, obviously FNA, you saw a much higher development of adenomas in the FNA C2. Again, this particular type of mouse tends to develop adenomas anyway. So you do see, sort of a non-zero control, and even FNA1, C1 was also non-zero, but C2 still increased it, as you might expect. So really, what we're looking at is it engineers a cancer-friendly environment. Now, how does it do this? And it really is its ability to manipulate its surrounding. It's the ability of not just to form a structure that leads to enhanced growth, uncontrolled growth, and the various qualities that then lead to tumor generation and some of the other negative effects. And it does this by essentially terraforming the gut and changing its metabolic outcomes. Now, what it was able to do is increase metabolic stress in that area. And it essentially is an oxidizer among other properties. Now, again, this doesn't just create free radicals, but essentially does create stress within its surroundings, stress within the immune system away from it. Now, inflammatory environment's not the whole story, but it's sort of a trickle-down effect of its effect living and colonizing the gut. Your body does have a very strong reaction to it. So you're going to obviously see cells that increase cell proliferation around this and its ability to affect those cells. And it really is, where it doesn't just arrive the gut, but it creates a specific niche that will subsequently develop cancer. So you talk about stress, oxidative stress, inflammation, affecting the host environment. It does have anti-immune qualities. And again, it's not just seems to be present, but it is, has an ability to lead to these changes that essentially cause mutations down the road within our cells and aggressive growth of our own cells, possibly as they try to combat this aggressively growing bacteria. And really the conclusion is, how do we fundamentally reframe this? This was really one of the first bacteria that's been really strongly linked. We do see bacteria from the mouth that end up in the heart. They end up all over the place, but this one seems to be growing. And it begs the question, are there various ways, tools that we use in the mouth, like inhibiting corium sensing, that maybe could play a role in the gut and in other types of areas where we're seeing bacteremia and bacterial growth and its negative effects. So a lot of research to be done. I think we're just barely scratching the surface. Again, we're talking about an astronomically high number of microorganisms that live in the mouth, and how many of them have similar effects. And it's hard to say, and we may never quite understand. But the understanding is that there is a connection between the oral cavity and the gut, which you all know, but organisms can pass from the mouth to the gut and presumably pass back if you don't wash your hands. So it really does play a narrative and may change your overall understanding, where you can have things, especially if you have a large amount of bacteria in your mouth, you may have a substantial count of pathological bacteria in your mouth. It can transmit to the stomach. It can survive. It can colonize. It can live and thrive in the gut. And then it can ultimately, in its quest to survive and reproduce, it can cause a similar effect within the immune system around it, which can lead to cancers and incredibly pathologic states within the body. So Anyway, so what do we do? You know, what does this mean for oncology? It may mean that a large part of it is trying to prevent this specific bacteria from doing exactly what we're doing. Do we use stool to screen this? And perhaps there are options where if you're able to document and sift through this incredibly large amount of data, we have genomic data, we have protein data that were recently synthesized a few years ago. We have just an incredible amount of information that a human can't really sift through, and there's from different sources. Perhaps there's a way to go through and better predict other types of bacteria that do fairly well. And it also allows you to look at various therapeutic approaches. Are we looking to target C2 specifically? Can we do antibiotics that really target this clade? Maybe how sticky it is, its ability to traverse the stomach. What types of things can we do both orally and in the gut to help combat it? Can we find it sooner in people? That maybe there's certain vaccines you can give against this, and really trying to outsmart this type of things. And you got to understand, in the mouth, if you just try to kill everything, you may end up having an overgrowth of certain things. So you would want to specifically target this type of bacteria. And really what this means for you is, I think it does, understand that there is a connection there on a simple level. The bacteria in your mouth can and often does play a role. And hopefully this helps people understand the development of certain types of disease. And hopefully we can combat it a little bit better. So remember, oral hygiene, we always talk about it. I think beyond just improving your mouth, improving the overall quality of your oral hygiene can certainly help. You can drastically lower your bacterial counts. And it's not just about keeping your teeth clean, as this shows. Yeah, it can actually help in other ways and lower other risks. The studies aren't necessarily there, but the rationale, I think, is very strong to getting regular cleanings and keeping up on your oral hygiene. And I'm going to go ahead and say it, hand washing. I think a lot of people skip it. I think in this light, it does sort of say, well, hey, you probably got to really keep your mouth clean. And I always advocate, if you're about to brush your teeth, you might as well wash your hands first. If you're about to floss, just wash your hands first and then floss. I think it's a no-brainer. A lot of people skip it. And, the idea is that, fundamentally, circling back, it's a two-way street. So a lot of things that are going to help your overall health involve taking care of your oral hygiene as well. It's the belief that you can make a difference and you can improve your health with small steps is probably more beneficial than those small steps themselves. And, take care of your health, go to the doctor, go to the dentist, do those little preventive things, eat well, exercise, because that's all going to play a role. And you can sort of see how those things all add up. Not smoking, not drinking too much, are going to play a role. And that's going to lower overall your cancer risk indirectly, because you're just taking care of it. You're going to lower the bacterial counts in your mouth. You're going to be healthier. Your immune system is going to be better. And you can start to see how these simple actions really play a role. Anyway, sources, there's many more than this, but these are the big ones. Obviously, this is going to be turned into a podcast of Dr. Joshua Grenier, and then we're also going to put it online. Again, we're going to be doing these around 6 a.m. every day. So we'll set up a schedule soon. All right, take care.